In this video, we'll discuss innate immunity, but a few important exam questions before we start the topic. Question number 1. How do eosinophils terminate inflammation? Number 2. Which cell type matures in the bone marrow? Number 3. What are the examples of macrophages in different tissues? Number 4. Which blood cells have large nucleus and large granules? Number 5. Which complement factor is the key factor and why? And number 6. What inhibits protease activity? Now, innate immunity, which is non-specific. Innate immunity provides first defense against pathogens. It recognizes pathogens by number 1. hematopoietic cells or blood cells, and bone marrow gives rise to all hematopoietic cell types. And number 2, by non-hematopoietic cells, and they include number 1, skin, mucosa, temperature, pH, and body secretion. For example, HCl in the stomach, saliva, and tears. And number 2, complement cascade, cytokines, and antimicrobial peptides leading to cell lysis. Body tissues that recognize pathogen: number 1, phagocytic and NK cells, natural killer cells, and number 2, complement. Now, phagocytic cells. These includes neutrophils, eosinophils, basophils. They are found in the circulation. Number 2, monocyte and macrophages. They are found in the tissues. Monocytes migrate from circulation to the tissue. And number 3, dendritic cells and epithelial cells. They are found in the tissues. Now, phagocytic cells of the innate immunity, they include granulocytes. They are neutrophils, eosinophils, and basophils. Granulocytes are derived from the stem cells in the bone marrow and found in the circulation. They are present in all forms of inflammation and innate immunity. And what's chemotaxis? Chemotaxis is bacterial product. They attract neutrophils to these infected area. This is known as chemotaxis. These cells can lead to host tissue damage and systemic necrotizing vasculitis. Now, neutrophils. Neutrophils are multi-lobed. They have 3-4 lobes, and they are the first cell to come at inflammatory sites. Neutrophils phagocytose and kill the bacteria by granules. Which granules? Number 1, AzuroPhilic granules, and the other granules. So what AzuroPhilic granules contain? They contain myeloperoxidase, lysosomes, and elastase, whereas the other granules contain lysosomes and collagenase, and superoxide radicals are generated from them. The superoxide radicals cause direct injury to the cells. Neutrophils express FC receptors for immunoglobulin G and activate components of the complement C3b. Now, eosinophils and basophils, they are bilobed cells and can also lead to host tissue damage and systemic necrotizing vasculitis. Eosinophils also express FC receptors for IgG, and they are cytotoxic to many parasitic organisms. So eosinophils are bilobed and they can also lead to host tissue damage and necrotizing vasculitis, and they also have FC receptors for IgG like neutrophil, and they are cytotoxic to parasitic infections. And also, eosinophilic neurotoxin damages the tissue and causes hypereosinophilic syndrome. Now, how do eosinophilic granules stop inflammation? The eosinophil contains anti-inflammatory enzymes, histaminase, aryl sulfatase, and phospholipase D that inhibit inflammation. The basophil and the tissue mast cells contain cytokines, interleukin 4 and 6, and TNF-alpha and interferon gamma. So basophil contains interleukin 4-6, TNF-alpha, and interferon gamma, and can respond to bacteria and viruses. They also binds to antipathogenic antibodies and protects against parasitic diseases. They also respond in allergic and inflammatory diseases. They have receptors for immunoglobulin E and release histamine, and eosinophilic chemotactic factors for anaphylaxis and neutral proteases. They have surface receptors for activated complement C3a and 5a, so they are active in allergic and inflammatory diseases also. Now, mast cells. Mast cells occur in tissue and epithelia. They have a small nucleus with large blue granules. The other cells that have blue granules are basophils, and mast cells contain histamine and actin allergy, but basophils have bilobe nucleus. Now, monocytes and macrophages. Common features of monocyte and macrophages are that they kill the microbes through the release of hydrogen peroxide and nitric oxide, and inflammatory mediators from macrophages cause destruction of antibody-coated bacteria and tumor cells. Monocytes and macrophages are the major antigen-presenting cells, APC, and they have receptors for the Fc regions of IgG, and they activate complement and various cytokines. Now, monocytes, as I already explained, like macrophages, they produce hydrogen peroxide and nitric oxide. They also arise from the bone marrow. They are large cells. They have large, round, heart-shaped nucleus with scanty cytoplasm. Monocytes follow neutrophils in the inflammatory area, so they are second cells to come at inflammatory site, and they migrate from circulation to the tissue and phagocyte bacteria, foreign bodies, and the dead cells. And in the tissues, monocytes are converted into macrophages. Now, macrophages. Macrophages are not found in the blood, but they are found in the lymph nodes, spleen, bone marrow, tissue, serous cavities like peritoneum and pleura. And the examples of macrophages are alveolar macrophages in the lung, kupffer cells in the liver, osteoclast in the bone, and microglia in the CNS. So, alveolar macrophages, kupffer cells, osteoclast, and microglia cells in the CNS are macrophages. Like monocytes, they also produce hydrogen peroxide, nitric oxide, and phagocytose and kill the bacteria by producing antimicrobial peptide and cytokines, interleukin 1, 6, and 12, and TNF-alpha, chemokines, leukotrienes, prostaglandins, and platelet activating factors. Activated macrophages mediate lytic activity and kill tumor cells in the absence of antibody mediated by cytokines, that is TNF-alpha and interleukin 1. So the macrophages have lytic activity and kill tumor cells in the absence of antibodies by TNF-alpha and interleukin 1. Macrophages ingest bacteria and virus infected cells and undergo apoptosis. And the macrophages that are damaged by intracellular infections are phagocytosed by dendritic cells. So the fate of the macrophages is that bacteria and virus infected cells or macrophages, they undergo apoptosis, and the macrophages that are damaged by intracellular infection are phagocytosed by dendritic cells. Now, the dendritic cells. Dendritic cells are the most potent APC, antigen presenting cells. They have long cytoplasmic arm like a spider angioma. So cells with long cytoplasmic arm are the most potent APC presenting cells. The dendritic cells are important bridge between innate and adaptive immunity, and the two types of dendritic cells are myeloid and plasmacytoid cell. Both cells produce chemokines that attract neutrophils, B cell, T helper, and T memory cell. So dendritic cells are a bridge between innate and adaptive immunity. They have long cytoplasmic arms and they produce chemokines that attract neutrophil, B and T helper and memory cell. The myeloid cell type of dendritic cells are of two types, interstitial and Langerhans. The interstitial cells produce interleukin 10 and 12, and Langerhans also produce interleukin 12 in both circulation, tissues, lymph nodes, skin, epithelia, and thymus. The other property of myeloid cell is that they can differentiate into monocyte macrophages and tissue-specific dendritic cells. So myeloid cells are a type of dendritic cells which can differentiate into monocyte macrophages and tissue-specific dendritic cells. Now the other type of the dendritic cell, plasmacytoid cells, they have less antigen presenting cells, APC, and they look like plasma cells. The plasmacytoid cells are in the circulation and lymphoid organs and produce interleukin 1 and interferon alpha against viral infection. So interferon alpha has anti-tumor and antiviral effect. They kill pathogen infected cells, activate T cells to form killer T cells, and activate NK cells of the innate immunity. So plasmacytoid cells produce interferon alpha that kills anti-tumor antiviral cells, kill pathogen infected cells, and activate T cell to form killer cells and activate NK cells. The dendritic cells, when come in contact with viral proteins or host cells, release cytokines and chemokines, attract neutrophils, B and T helper and memory cells, and recruit B and T cells of adaptive immunity. Thus, dendritic cells are important bridges between innate and adaptive immune system. So, dendritic cells, when come in contact with bacteria, virus, or host cells, release cytokines and chemokines and attracts neutrophils, B and T helper and memory cells, and recruit B and T cells of adaptive immunity. Now, natural killer cell or NK cell. NK cells arise from both bone marrow and thymus and have large granules. So they are the T cells with large granules and constitute 5 to 10% of the peripheral blood cells. The NK cells are non-adherent, non-phagocytic cells and are non-immune cells. That is, they never came in contact with the target cell before and cause non-antibody mediated killing of target cell. So they kill the target cell even if they don't come in contact with the antibodies previous. They can kill the malignant cells, foreign cells and viral infected cells. How do they kill it? They kill it with large AzuroPhilic granules. The NK cells produce and are activated by interferon, and they have the receptor for Fc portion of immunoglobulin G. They also have features of neutrophils and monocyte macrophage. So they have features of all the three cells: neutrophil, monocyte, macrophage. So they mediate antibody dependent cellular cytotoxicity. NK cells may have different types of receptors are killer immunoglobulin-like receptor KIRS that inhibit and killing of HLA positive cells. NK cells inactivation by KIRS is central mechanism to prevent damage to the normal host cell. So killer immunoglobulin receptor prevent normal host cell damage. Number 2, NK cells have natural cytotoxicity receptors that kills the target cell. And there are two other receptors on the NK cell, activators and inhibitor receptors of the NK cell. Inhibitors of the NK cell don't respond to uninfected and non-malignant cells, and activators of the NK cell attack malignant and virally infected cells. So different types of NK cell receptors are killer immunoglobulin-like receptors which prevent the damage to the normal cell, and natural cytotoxicity receptors that kills the target cell. The inhibitors of the NK cell don't respond to uninfected and non-malignant cells, and activators of the NK cell which attack malignant and virally infected cells. The other function of NK cell is that it damages intracellular bacteria. For example, Listeria monocytogenes and Mycobacterium tuberculosis. They are also attacked by NK cells. The functions of the NK cell is hyper-responsive in which disease? In chediak higashi syndrome with defective degranulation of the neutrophil lysosome. So summing up, the NK cells that they form 5 to 10% of the peripheral blood, and they are non-immune, non-phagocytic, non-adherent and non-immune. That non-immune means they attack the cells which do not have antibody, and they attack malignant cells, virus infected cells and foreign cells with their AzuroPhilic granule, and they have features of neutrophils and macrophages and monocytes, so they mediate antibody dependent cellular cytotoxicity. Different types of NK cell receptors are killer-like receptors, natural cytotoxicity receptors, inhibitors of NK cells and activators of NK cells. They also attack some intracellular bacteria that include Mycobacterium tuberculosis. Now, complement system. Complement system is a system of soluble components of innate immunity. It consists of C1 to C9, 9 protein enzyme which enhance inflammation. Enzymes of the complement system are serine. So as a result of antigen-antibody combination, there is cell lysis, and this is enhanced by complement system. So what happens in complement? WBCs are attracted to the site of the complement system, and pathogens are phagocytosed by neutrophils and monocytes and macrophages. Complement activation is a critical component of innate immunity. 4 pathways of complement. Number Number one, classic activation pathway, which is dependent on antigen-antibody reaction and is activated by C1, 4 and C2. Alternative pathway, antibody independent, activated by microbes or tumor cell by binding of C3 directly to the pathogen. For example, in immunoglobulin A nephropathy, IgA activates alternate complement pathway causing glomerular damage. Number 3 is the activation pathway activated by microbe, and number 4 terminal pathway, a combination of the above three that leads to cell lysis. So the complement system consists of 9 enzymes from C1 to C9, and the key factor is C1, which has 3 subunits, C1q, C1r, C1s, in both innate and adaptive immunity. So what's the function of C1? C1 recognizes bacterial and viral sugar structures which are different from a cell. Interaction of C1 with antigen activation produces holes in the antigen that leads to leaks in the cell causing cell lysis. C1 binds to the immunoglobulin that is bound to antigen. So it binds to the immunoglobulin that is bound to antigen, and it triggers a reaction that activates C3 and C5 and converts them into C3a and C5a, which are the active form, and that releases histamine from granulocytes, mast cells and platelet, leading to vasodilatation and increased capillary permeability. So the basic function is to increase capillary permeability by vasodilatation, and this is done when C1 binds to antibodies, immunoglobulins, which are already bound to antigen. That causes activation of C3 and C5 and converts them into C3a and C5a, and that releases histamine from granulocytes, mast cells and platelet, leading to vasodilatation and increased capillary permeability. C3b causes opsonization, and C5a, C5b, C6 and C7 are chemotaxis and attract WBC at the site of antigen-antibinding reaction. C3 cleavage by first 3 pathways activate C5, C6, C7, C8 and C9 that leads to cell lysis. So complement is a system of soluble protein of innate immunity that has serine protease. When antigen combines with antibody, to this antibody the complement attaches, and that attracts the WBCs, and pathogens are phagocytosed by neutrophil, monophage and macrophages. There are 4 paths. Number 1, classical activation pathway, antigen-antibody dependent. Alternative pathway, antigen independent. Activation pathway activated by microbes, and the 4th pathway is a combination of the 3 that leads to the cell lysis. The first step or the key factor in the complement is C1, which recognizes the bacterial and viral sugar structures, and C1 combines with the antibody that is attached to the antigen and makes holes in the antigen by causing chemotaxis and attracting neutrophils, monophage and macrocyte at the cell. C1, when combined with immunoglobulin, activates C3 and C5, and that releases histamines from granulocytes, mast cells and platelet and causes vasodilatation and increased capillary permeability. Now these enzymes, as I already told, have protease. So what substance inhibits protease activity? C1 esterase inhibitors inactivates protease activity of C1 in the classical pathway, and C1 esterase inhibitor deficiency causes hereditary angioedema. Now how the complement is tested? Decrease in C2 and C4 indicates activation of the classical path, and decrease in factor B indicates activation of alternative path, and decrease in C3 indicates activation of either. So decrease C2 and 4 indicate classical pathway activation, decrease factor B indicates alternative pathway activation, and decrease C3 indicates activation of either system. Now answers to the question, how do eosinophils terminate inflammation? The eosinophils have certain substances like histaminase, aryl sulfatase and phospholipase D which terminate inflammation. Question number 2, which cell type matures in the marrow? Cell types that mature in the marrow are granulocyte, monocyte, dendritic cells, T lymphocytes. So the T lymphocytes are not matured in the bone marrow. What are the examples of macrophages in different tissue? Alveolar macrophages in the lung, kupffer cells in the liver are macrophages, osteoclasts in the bone, and microglia cell in the central nervous system are the examples of macrophages. Which white blood cells have large nucleus and large granules? The answer answer is NK cell, important in innate immunity. Which complement factor is the key factor and why? The key complement factor is C1, which has 3 subunits in both both innate and adaptive immunity, and it's important because it recognizes viral and sugar structure to begin the process. Question number 6. What inhibits protease activity? C1 esterase inhibitors inactivate protease activity of C1 in the classical pathway.